Sci-Fi, we're going to zoom in on this brutal, beautiful, and metal biology and really extract the mechanisms that underpin this extraordinary struggle for survival that ultimately results in the most magical thing, new life.
Right. But you have to understand the paradox here, because it's everything.
It is. The vulnerability of this new life, I mean, a tiny clump of cells fighting for every single hour of existence, is matched only by its immense biological aggression and sophistication.
That's an energetic drain,
Yeah.
a physical challenge.
And a severe multi-month test of the mother's entire systemic balance. So we're starting our deep dive right at the beginning.
Let's do it.
Setting the stage for the very first battle, the incredible and deadly selection process that happens long before that new life has even formed.
Okay, let's unpack this opening scene. It's like the opening shot of an epic war film, but instead of 50,000 troops storming a beach, we have an army of tens of millions of hopeful invaders. The sperm cell.
Tens of millions. It's an enormous initial investment, and immediately they face death.
Right away.
The sources make it clear they are on an immediate tight timer. A sperm cell is essentially just a nucleus with a limited, pre-packaged fuel source.
Mitochondria.
Exactly, mostly concentrated in the midpiece. If they don't reach their goal fast, they simply run out of chemical fuel, their tails stop whipping, and they die.
And the female reproductive tract is not a welcoming highway. The source is painted as a fully operational fortress, deliberately set up with deadly barriers.
A fortress is the perfect word.
So why? Why is the mother's body so actively hostile to these potential invaders?
That's a crucial question. The body sees anything introduced into the reproductive tract as a potential pathogen or foreign body.
So it's just defense.
It's pure defense. The environment is engineered to kill almost all of them, selecting only the statistically strongest and fittest single cell. This is intense natural selection operating on a microscopic scale.
It's making sure that whatever gets through is robust.
Metabolically robust, exactly.
So if we're navigating this fortress, what's the first major deadly obstacle the Army encounters?
The first and maybe most famous is the acid test.
The acid test.
Yeah. The initial entry points, the vagina and cervix, are highly acidic. And this acidity is maintained primarily to kill harmful bacteria and protect the entire system from infection.
But that means it kills millions of sperm, too. It's just collateral damage in the name of safety.
Precisely. This environment is guarded by hundreds of thousands of specialized guard cells, and the acidic environment acts as a mass killer. It immediately kills millions of sperm.
Millions.
Especially the weaker, poorly formed, or slower ones, all within the first half hour of the journey. It's an overwhelming attrition rate right out of the get.
Wait, okay, if the body maintains this acidity for pathogen defense, how do any sperm survive at all? Do they have some kind of, biological armor?
They have a countermeasure, but it's provided by the partner.
Oh, okay.
The seminal fluid itself is highly alkaline.
So it's like a shield.
Think of it as a temporary chemical shield.
Yeah.
It momentarily neutralizes or buffers some of that intense acidity, creating a small protective microenvironment.
So it's a buffer zone.
It's a critical mitigating factor. It offers a crucial percentage of the Army just enough time to push through that initial assault into the less acidic cervical area. Without that alkaline buffer, the journey would be, a non-starter.
So the survivors, this tiny fraction that navigated the acidic gauntlet, they pushed through into the cervix. And this is often described as the second deadly stage, a true biological dungeon maze.
It is a treacherous maze, primarily characterized by thick cervical mucus and complex anatomical folds.
So like protein nets.
Exactly. Think of dense, complex protein nets. This is the ultimate physical obstacle course.
What's the specific mechanism of the cull here? How does it kill them?
It's filtering, purely based on motility and morphology. The mucus acts like a physical sieve.
Right.
So millions more find a brutal end. They get physically stuck, tangled in the dense glycoprotein matrix, or they get lost because they lack the smooth, straight motility required to navigate the channels.
So if you can't swim straight, you're done.
You're done. This process is so effective at weeding out abnormal or slow cells that it's estimated that the numbers drop from millions to just hundreds of thousands,
Right.
just crossing that cervical barrier.
But this is where timing becomes everything, right? The fortress occasionally lowers the drawbridge, offering what our sources call a ceasefire condition.
This is the critical timing element governed by the menstrual cycle. If the woman is ovulating, the body switches from pure defense to selective assistance.
Selective assistance, I like that.
The cervical mucus dramatically changes consistency. It becomes thinner, more watery, and it forms these organized channels, almost biological pathways, that actively guide the stronger sperm toward the uterus.
So the body's saying, fine, the defense budget is temporarily lowered, but only for the best of the best.
Exactly. The environment becomes a bit less hostile overall, which shows just how selective the mother system is. And get this.
What?
The act of passing through the cervix and uterus initiates a process called capacitation in the sperm.
Capacitation.
It's a final, necessary conditioning step where the sperm's outer membrane changes, making it chemically capable of breaking through the egg's protective layers. The cells that fail to capacitate are selected against, even if they reach the egg.
So that adds yet another layer of required transformation.
Yeah.
It's not just about surviving, it's about changing.
It is.
Okay, so we started with tens of millions. After the acid, the maize, the chemical changes,
Yeah.
how many are left to enter the final stage, the uterine cavity?
It's mind-bogglingly low. We're talking a couple hundred sperm.
A couple hundred.
That's often less than 0.00001% of the original army that actually passed through those initial tunnels.
That is statistically negligible. It's a rounding error.
It is. And the uterine cavity presents its own challenges. It has two tunnels branching out, the fallopian tubes, and the sperm have to choose the correct one.
The one with the egg.
The one leading to the egg. Here, the sheer distance, combined with the action of cilia that often sweep against the sperm's movement, calls millions more. The mucus in the tubes helps transport stronger sperm while weeding out weaker ones who can't maintain the pace or trajectory in this final stretch.
And if they take the wrong turn or lag behind, I understand the mother's dedicated cleanup crew arrives.
Oh, yeah. The mom's immune cells, specifically macrophages, are actively picking off and devouring every single cell that trails behind, takes the wrong turn, or shows any signs of distress.
So it's total cleanup crew duty.
It is. They're making sure no unnecessary biological material remains that could trigger a systemic immune response or an infection. The goal, the final target, is that egg waiting at the end of the correct tube.
So we've gone from 50 million to maybe 50 to 100 cells making it to the final rendezvous.
If that.
The scale of the attrition is just the most profound lesson of this first battle. And when they finally arrive, they face what you describe as the mighty giant egg.
It truly is an industrial-scale monster compared to them.
How so?
The egg is the largest cell in the human body. It is physically 10,000 times bigger than a sperm cell. And it's not passive, it's a massive resource repository.
And its resources are the key to the next phase of survival, right? It's not just about size, it's stored power.
Exactly. It is loaded with nutrients, proteins, and lipids to sustain the new life for days until it can implant. And crucially, it's loaded with mitochondria.
The powerhouses.
The powerhouses.
Just how loaded are we talking?
Nearly 100,000 mitochondria. To put that in perspective, an average somatic cell, a regular body cell, has about 2,000.
Wow.
This is 50 times more power storage than average. And this massive energy supply is absolutely critical because the fertilized egg, which we now call a zygote, has to rapidly divide multiple times, travel down the fallopian tube, and start signaling, all before it ever reaches a reliable external energy source.
So the egg is not easily conquered. The final act of this initial battle is a selective decision by the egg itself.
The egg allows the survivors to get close, but it's armed with layers of defensive proteins, like the zona pellucida. Once a single successful sperm manages to penetrate and merge its genes, the egg immediately triggers what's called a cortical reaction. It hardens those outer layers and ensures no other sperm can get in.
And this is the moment of transformation.
This is it. The genes of two individuals merge, and crucially, the resulting zygote immediately stops being genetically or immunologically part of its mother.
It is something else now, a new entity.
It is a new being with its own biological agenda. It's the potential for a human, but the mother's body hasn't decided if she wants to allocate resources to it yet.
So the fight isn't over.
The fight continues immediately, but the focus shifts from physical assault to sophisticated chemical diplomacy.
Which brings us smoothly to Section 2, the negotiation and the guardian angel clone.
Right.
We have this brand new entity, a rapidly developing cluster of cells, which immediately have to start this intensive, critical dialogue with the mother's body to gain permission to implant and survive.
And those initial critical days are spent in hyperspeed growth and specialization. The new life divides, going from 1 cell to 2, then 4, 8, and so on, all while traveling down the fallopian tube toward the uterus.
And by the time it gets there,
By the time it reaches the uterus, it's a couple of hundred cells strong. It's now known as a blastocyst.
and immediately, the cells specialize into two dedicated teams. One team will become the embryo, eventually, the baby, but the other team is the unsung hero, the biological infrastructure specialists.
These are the trophoblasts.
Crophoblasts.
They are fundamentally self-sacrificing cells. Their sole job is to turn into the placenta. This is the key. They are creating a temporary organ inside the mother's body that will make pregnancy possible.
And then, after birth,
They will be ejected and die.
this gives us that fascinating and profound concept of the guardian angel clone.
Think about that for a second. When you were just a tiny cluster of cells, a whole other part of your genetic self existed.
Not a twin.
Not a twin that would become another person, but a distinct set of cells, a clone, if you will, whose only purpose was to ensure your existence. They handled the aggressive infiltration, the chemical warfare, the resource theft.
And they never got to become a person.
They never had the chance to become a human themselves. They were entirely dedicated to infrastructure, defense, and survival.
That is an incredible biological sacrifice at the cellular level.
Yeah.
But despite this initial organization, the following days are the most dangerous period of this whole sci-fi epic.
Absolutely. We're talking about the period before successful implantation.
And it's incredibly vulnerable.
Immense vulnerability. The source describes the movement like a tennis ball rolling through syrup, or a tiny boat adrift in a very large sea. It's precariously moving, trying desperately to take hold of the uterine wall.
And if it fails?
If it fails here, the mother's body will simply sweep it away. This is where the intense chemical dialogue, the negotiation, begins.
The embryo has to announce its presence and make its pitch immediately, right? Releasing dozens of chemicals to announce, I am here and I am viable.
It's an urgent, insistent plea. It's often called the maternal recognition of pregnancy. The embryo has to convince the uterine wall, the endometrium, to switch from its default mode of preparing for menstruation to a supportive mode.
The receptive mode.
A receptive mode called desiderialization, where it thickens and becomes welcoming.
So what is the primary chemical argument the embryo is making? How does it plead its case?
The loudest chemical announcement, the main argument, is the release of massive quantities of human chorionic gonadotropin, or HCG.
Ah, the one from pregnancy tests.
The very one. And the purpose of HCG is twofold. First, it tells the mother's body, specifically the corpus luteum and the ovary, do not die, keep producing progesterone.
And progesterone is the key.
Progesterone is the master hormone required to maintain the uterine lining.
So if the embryo doesn't yell loud enough, that corpus luteum dies, progesterone drops, and the lining is shed, taking the embryo with it.
Precisely. If the embryo's chemical conversation, its HCG signal, is too weak or comes too late, the mother's body initiates the menstrual cycle and the pregnancy is lost.
And that chemical conversation triggers a response.
It triggers a response of dozens of counterhormones and immune signals back from the uterus, which essentially says, we hear you. Now let's assess your quality.
This assessment phase is where we see the divergence of interests solidify, because the mother's body is fundamentally a shrewd negotiator focused on self-preservation and resource management.
Absolutely. From an evolutionary perspective, pregnancy is the single largest energy investment a female mammal can make.
So we can't afford to waste it.
Right. If the mother's body is not satisfied with the quality of the chemical conversation, if the HCG signals indicate the embryo is low quality or metabolically robust or is simply growing too slowly, the uterus will not allow the desiderialization process to complete.
And implantation fails.
Implantation fails. It's a pragmatic biological veto designed to prevent wasted energy.
The mother's body is performing a quality control assessment that determines life or death.
Yeah.
But if the embryo is rejected, does the mother even know a rejection took place?
Often not. No. These very early losses, before implantation is secure, are often chemically undetectable and manifest as a slightly late or heavier period.
Wow.
But for the embryo, the stakes are existential. This is a life and death situation, and the embryo will try to stay alive at any price.
And if that polite chemical conversation, the HCG pitch, isn't working fast enough,
The tactics must escalate.
which brings us squarely into section 3, the aggressive phase. Parasitic infiltration. The embryo stops politely asking and starts taking direct, invasive action.
The language is indeed perfect. Once it gets close to the uterine wall, if the endometrium hasn't fully committed to the decidual change, the embryo stops asking politely and deploys thousands of infiltration units.
Infiltration units. What exactly are these units on a biological level? We hear terms like human virus, but what are we looking at?
We're looking at specialized tiny messengers called exosomes and microvesicles. They are bubbles filled with genetic material, specifically microRNA and signaling proteins released by the trophoblast cells. And they're described as kind of like a human virus because their goal is infiltration.
They invade.
They fuse with the mother's uterine cells and chemically and genetically invade them.
So the goal is to brainwash the mother cells. How successful is this reprogramming?
The mission is to reprogram the maternal cells so that they actively help the embryo attach and remodel, rather than continuing to reject or ignore it. They change the maternal cell's gene expression, encouraging them to release pro-attachment growth factors and suppress local immune responses.
It's a sophisticated biological subversion to secure the beachhead.
It really is.
Before the full aggressive invasion begins, the embryo, having used up most of the egg's power reserves, needs a major pit stop.
This brings us to one of the weirdest features of this phase: the provision of uterine milk.
Uterine milk.
It's the perfect analogy, but yes, it's not real dairy. The technical term is histotroph.
What's in this histotroph, and why is it so crucial?
It's a clear fluid secreted by the uterine glands, filled with essential high-energy nutrients like glycogen, lipids, and key hormones and growth factors.
And the embryo just soaks it up.
The young embryo sucks up hungrily this fluid to gain additional energy. It's a critical pre-implantation fuel stop. It needs this energy surge because the egg's mitochondria alone cannot sustain the necessary rapid cell division and the chemical signaling required for implantation.
So it's fueled up, it started the brainwashing process. The final target is the mother's blood supply, and that requires a move far more violent than chemical persuasion.
This is the critical transition where the peripheral layer of trophoblasts specializes into the syncytial trophoblast, what the source refers to as the violent invader.
Describe this invader. What makes it so aggressive?
It's often visualized as a tiny parasitic octopus with many arms. The syncytial trophoblast is a massive, multinucleated cell layer that acts like a biological drill. It begins drilling directly into the uterine tissue, spreading and growing rapidly.
And its orders are brutal, literally ordering the mother's cells to die.
Yes. This phase involves a command structure that triggers apoptosis-programmed cell death in the mother's uterine cells to clear a path.
So it's clearing the way.
It kills others directly via chemical dissolution, or the cells literally devour some maternal cells whole to access their nutrients. The point is survival at all costs, requiring the systematic destruction and remodeling of the immediate maternal environment to access the blood vessels.
That sounds genuinely horrifying, yet the sources emphasize that this process is highly regulated. What prevents this aggressive infiltration from damaging the mother permanently?
That's where the high regulation comes in. The mother's decidualized tissue is not passive. It puts up a counter-defense, limiting the depth and breadth of the invasion.
So there's a push and pull.
A constant push and pull. If the invasion is too shallow, the pregnancy will fail due to lack of resources. If it is too deep or uncontrolled, a condition that can lead to pathologies later, the maternal tissue attempts to contain it. The entire process serves as a highly refined biological test.
Only the best invaders make it.
Only the embryos capable of achieving a perfectly regulated, aggressive invasion are allowed to continue.
And this leads us to one of the most sophisticated quality control mechanisms we've discussed yet. Something the mother's body monitors constantly: metabolic noise.
Yes, the mother's body is a master of biochemical eavesdropping. It is constantly observing the embryo's metabolic activity.
Like how much energy it's using.
How much energy it's using, how fast it's growing, and the chemical signatures it releases. This is the ultimate non-invasive health check performed by the surrounding maternal immune and stromal cells.
So what does a flawed embryo look like metabolically? What is the flaw trap?
It looks noisy. If the embryo has significant genetic damage or chromosomal abnormalities, it spends a disproportionately large amount of energy on repair mechanisms.
Trying to fix itself.
Unfolding and refolding damaged proteins, attempting DNA repair, and growing erratically. This inefficiency results in the release of a load of noisy chemicals, distress signals, inflammatory cytokines, and waste products.
And that noise is picked up by the mom's immune system.
Immediately. The mother's immune cells, particularly the uterine natural killer cells, pick up on this metabolic distress. This noise is interpreted as non-viability or a potential energy sink.
So it's a bad investment.
It makes it significantly more likely that the immune cells will destroy the embryo because the mother's body recognizes the energy investment is unlikely to yield a healthy return.
It's the ultimate cost-benefit analysis. But then there's the opposite problem, the weak trap.
Mm,
being too quiet.
exactly. If the embryo is too weak, maybe struggling with cellular division or resource uptake, it is metabolically too quiet. It's not generating the robust chemical signals.
Not enough HCG.
Not enough HCG or growth factors required to trigger the full supportive decidual response in the mother's tissue. In this case, the mother's body interprets the silence as failure, stops communicating with it, and the pregnancy ends.
So the embryo must hit the just right zone. Strong enough to grow aggressively and loudly announce its presence, but not so flawed that it's metabolically noisy with distress signals.
And when the embryo is metabolically just right, the mom cells respond with a flood of supportive chemicals, vasodilation factors, growth hormones to support its continued growth.
Which sets the stage for the next incredible paradox: how this foreign entity survives the mother's systemic immune response.
Right.
This is truly the most astonishing part of the entire journey. The immune system is designed to kill anything non-self, and this embryo is definitively non-self. It's carrying half the father's genes.
It is.
How does the mother's body override millennia of evolutionary programming without completely compromising her own immune defenses?
It's section 4, immune manipulation. The mother's immune system is not turned off.
It's not.
It is entirely retasked for protection. The uterine immune cells, especially specialized immune cells called UNK cells, uterine natural killer cells, surround the embryo and actually start helping it.
They establish the safe zone. So what makes this zone safe from the mother's own heavy artillery like T cells?
It is a physical and chemical safe zone maintained by the trophoblasts themselves. The trophoblast cells are chemically camouflaged. They express a non-classical major histocompatibility complex molecule called HLA-G.
HLA-G. What does that chemical do?
Think of it as a biological security badge that flashes friendly instead of invader. Regular MHC molecules tell T cells, "This is foreign, kill me."
Right.
HLA-G, however, interacts specifically with the mother's local UNK cells and T cells, and it instructs them to stand down. It's a chemical do not disturb protect this unit sign posted right around the developing life, preventing acute rejection.
That's brilliant evolutionary adaptation. But the embryo isn't foolish enough to rely solely on the mother's goodwill.
Absolutely not. The defensive trophoblasts maintain their own perimeter. They send out specialized signals, often in the form of certain soluble proteins, that actively kill the mother's immune cells if they get too close and risk breaching the protective zone.
So it's a dual defense strategy.
The mother protects it systemically, but the embryo maintains localized, deadly defenses just in case.
Here's where it gets fundamentally, permanently interesting. And honestly, this detail changes how we must view the biological connection between mother and child, potentially decades later. The concept of microchimerism.
This is perhaps the most surprising and profound detail of the entire deep dive. It is the permanent infiltration.
Permanent.
Hundreds, potentially thousands of cells leave the embryo entirely. They separate, move through the mother's blood system, and spread all across the mom's body, permanently infiltrating her organs.
Which organs?
Her bone marrow, her lungs, and yes, even her brain.
They just integrate into her permanent biological structure.
They migrate, integrate, and survive, sometimes for decades.
Why? What are they doing?
The primary hypothesis is that these fetal cells carrying the father's genes are communicating directly with the mother's systemic immune system, acting as a permanent cellular reminder. This embryo is acceptable, do not attack, maintain tolerance.
But the lasting nature of this is the truly mind-blowing takeaway. It means the physical presence of the child remains with the mother long after birth.
Indeed. These cells may stay inside the mother for years or even decades. The source material states it so powerfully. It is likely that parts of you are still in your mother.
So part of me is still inside my mother.
You, the adult listener, are not just genetically connected to your mother. You have physically left a small, living, self-replicating piece of yourself inside her long after you left the womb.
And research is now exploring the potential long-term effects of this. Is it purely immunological, or are these cells active?
There is fascinating ongoing research suggesting these cells might be active contributors to maternal health. For example, some studies hypothesize that fetal cells might migrate to areas of maternal injury, say a damaged heart or liver, and participate in tissue repair.
So they help heal her.
Effectively helping to heal the mother. This turns the initial parasitic relationship into a deep, two-way biological dependence that can last a lifetime.
While this systemic infiltration is going on, the construction crew, the remaining trophoblasts, is busy building the main habitat, the placenta fortress.
The construction trophoblasts build the placenta, A spongy, finger-like structure composed of villi that expand dramatically into the uterine wall. It's temporary, it's ejected and dies a silent death after birth, but while it exists, it is an enormous fortress and the primary life support system.
What's its most critical defense function beyond that HLA-G camouflage?
Protection from infection. The placenta acts as a sophisticated filter and barrier, guarding the fragile fetus from most microbes that circulate in the mother's blood. It even develops its own mini immune system.
A mini immune system.
Specialized placental immune cells that gobble up anything that poses a threat before it can reach the fetus.
And this fortress is also the central command in the ongoing resource war, which is essentially a conflict over fuel supply.
Absolutely. The process of tapping the maternal blood supply is where the aggressive nature of the trophoblasts reaches its peak. Other placental cells creep along the inside of the mother's spiral arteries.
The blood vessels supplying the uterus.
Correct. And they don't just tap them, they launch a full-scale takeover and remodeling.
What does that remodeling entail?
It's a physical conversion. The trophoblasts strip away the muscular walls of the mother's arteries and replace them with their own fibrous lining. This turns the arteries from narrow, high-resistance vessels that the mother's body could constrict
To save energy or control blood pressure.
into wide, low-resistance, permanently open funnels.
So the mother's body loses all control over the blood flow to the placenta.
Precisely. This ensures constant, maximum blood flow to the fetus, overriding any maternal physiological signals to conserve energy. Once the blood flow is secured via the umbilical cord connection, the placenta takes over the central command of resource distribution, prioritizing the fetus above all else.
And it does so ruthlessly, prioritizing growth fuel, specifically glucose.
The placenta releases a cocktail of hormones, notably human placental lactogen, HPL, which makes the mother's body resistant to her own insulin.
So her own cells can't take up the sugar.
Right. This ensures that the glucose circulating in her bloodstream stays there longer, effectively funneling it directly to the fetus. The placenta is actively stealing energy from its mom to fuel the explosive growth required.
And if the fetus is too aggressive, asking for too much energy, what are the systemic consequences for the mother?
If the fetus goes too far and asks too much, the extreme insulin resistance induced by the placenta can lead to gestational diabetes for the mother. Her body struggles to manage her own glucose levels because the placental demands are so immense.
It's effectively starving her own tissues of energy.
It is. And failures in that initial spiral artery remodeling process are strongly linked to serious complications like preeclampsia, highlighting how critical and potentially dangerous this balance of resource theft and maternal adaptation is.
This intense physiological conflict brings us to the most abstract level of the war, the deep genetic conflict playing out inside the fetus itself.
There is a fragile peace dictated by necessity. Inside the fetus, the genes inherited from the mother still retain a stronger evolutionary allegiance to her own long-term survival and health.
But the father's genes,
But the genes contributed by the father want the fetus to survive and grow at all costs, often pushing for faster, larger development.
this sounds almost intentional, as if different genes have different allegiances.
They do, due to a phenomenon called genomic imprinting. Certain genes are expressed only if they come from the father, and others only if they come from the mother.
So what's the difference?
Paternal imprinted genes often promote rapid, large fetal growth, like the insulin-like growth factor 2 or IGF-2,
Yeah.
while maternal imprinted genes often restrain growth, ensuring the mother is not consumed by the process.
So the two sets of genes are constantly engaged in a biological tug-of-war over size and resource allocation.
Their fundamental interests are not perfectly aligned, but they have to cooperate. The sheer energy demand, the fetus increases its weight over 1 billion times during gestation, requires a staggering amount of temporary cooperation to reach the goal of a viable birth.
And if they reach this final stage,
If they reach this final stage in the pregnancy, chances are extremely high. The result will be a baby, a proper human being, emerging from this epic biological war.
what an incredible journey we've charted. From a hopeless army of millions reduced to a couple of cells, to a full-scale biological fortress powered by permanent genetic infiltration. If you are alive today, you navigated and won an amazingly brutal, complex selection process.
It was a genetic journey that involved a deadly obstacle course where you were reduced to a tiny fraction of an army. It was followed by desperate chemical words spoken to the mother's cells and sneaky, almost parasitic actions designed to outsmart the biological system and ensure your survival. You were, in a very real way, born of aggression.
But that aggression is only one side of the story. We have to balance that inherent brutality with the concept of choice, of selection, and of profound maternal protection.
Absolutely. You were also chosen by the hormones that led you to the right place, chosen by the mighty egg that allowed you to merge and triggered the final selection process.
Right.
You were approved by your mom's body, which liked how you spoke to her chemically and metabolically, and you were protected by her cells, the UNK cells and the decidua that shielded and cared for you.
All nourished by an incredible amount of energy and bodily tissue that your mother gifted you.
Exactly.
It is the ultimate biological truth. Our biology is often brutal and unforgiving, governed by cold energy, physics, and ruthless selection, but it is necessary for the greatest wonder there is, the creation of new life, of all of us.
And if we look back at the timeline, we see that around 8 weeks after fertilization, the transition from embryo to fetus begins.
8 weeks.
At this point, the new life is about the size of an olive, and its organs start to form, turning it from a microscopic blastocyst into something visually recognizable and distinctly human-like.
Which leaves us with our final provocative thought, built directly on the complexity of this fluid, negotiated, and aggressive beginning.
Hmm.
If the process of becoming a new life involves so many phases of selection, genetic conflict, and biological negotiation, where exactly do we draw the line?
The entire journey is a fluid transition, an elegant continuum of development. We went from a single cell to a specialized clump of cells to a small, recognizable entity. Is there a clear, single scientific point where that clump of cells becomes a human being, independent of the resources it is currently stealing?
Not really.
Not really.
It's a continuum that every society, every legal system, and every person defines differently, be it morally, legally, or philosophically. The moment we mark that transition from potential to person is always marked differently, often arbitrarily, relative to the biology we just explored.
So consider this as you mull over your own aggressive, beautiful beginning. The complexity of your creation far surpasses any single definition. You are the result of a biological war, won by diplomatic skill, genetic infiltration, and finally, the ultimate act of maternal generosity. Think about where you draw the line and why that complex, hidden journey informs your answer.